Hi, I'm Jeremy. I'm a third-year grad student in Alex's lab at Ohio State, Nationwide Children's, and today I'll be talking about mapping variants from multiplex assays of variant effect, or MAVEs, to human reference sequences. So to provide some background, MAVEs stand for multiplex assays of variant effect. They are a technique that allow for the effects of thousands of variants to be investigated in parallel. Common examples of MAVEs include deep mutational scanning to measure protein variation and massively parallel reporter assays to measure regulatory variants. And these experiments basically yield a set of scores that describe the functional effects of studied variants. So we've seen the example in panel A. We have, before selection, 3 white, 5 blue, and 2 red, 3 white, 5 blue, and 2 red colonies. We impose some sort of selective pressure, and we get back 3 white, 2 blue, and 5 red. And by computing this ratio, we're able to essentially compute an enrichment score for all variants that we examine. And you can think about for panel B, if you examine across the entire sequence and do all possible variants for that given position, we can generate this plot where we have an enrichment score for every variant in every position. And this can be useful for producing functional effect data for variants that have not been previously examined in studies. So as I said, MAVEs can have potential clinical relevance as they can help interpret variants of unknown significance. So in the context of the ACMG guidelines, MAVE data could serve as PS3 evidence, as it could be well established in vitro and in vivo functional studies. So if you think about having this data available, you could previously classify a variant that was a VUS to a variant that has a more known classification of pathogenicity or being benign. So it could be clinically useful. In recent years, as more MAVE data has been kind of gathered, MAVEDB has emerged as a public repository for data sets from MAVE experiments. I have here kind of a toy example of a MAVE score set that you would see in MAVEDB. So there's a metadata field that includes a target name, such as a gene symbol, a type, so protein coding. It could be for a regulatory element or for other non-coding if that's not available. And then we include a reference sequence that describes variants that occur in that sequence. So we have a score matrix that has a substitution at position 1, position 2, and position 3, and an associated enrichment score for all variants in that position. And we can think about doing this for all different variants along the entire target sequence, so we can generate a pretty big data set if we have a target sequence that's over 1,000 proteins long or 1,000 nucleotides long or even longer. In MAVEDB, at the time of analysis, there were 299 total score sets. 209 described kind of human biology, so we looked at that for our analysis. And breaking down further, we had 168 protein coding score sets and 41 regulatory score sets. And of those 209 score sets, we had 176 DNA target sequences and 33 with protein target sequences. And there are different contexts for those experiments too. So the majority was human. We also had yeast experiments, bacteria, mice, and bacteriophage, and also some where the context was not available. So for our study, we wanted to basically take in this MAVE data and map it to human reference sequences to enable dissemination of this data to downstream precision medicine resources. So if you think about it, like on the left, we have different model organisms and different data that comes from these organisms, but it's not described with respect to a reference sequence. So it can't really be used for downstream clinical interpretation. So by mapping it and describing it with respect to a consistent human reference sequence, we're able to integrate this data into resources that kind of use that data, such as the USCSC genome browser or the ensemble variant effect predictor. Next, I'd like to go over the mapping methodology for how we kind of did this process. So there are 3 main parts. So after we determine that the score set is for humans, we first want to align the target sequence to the human genome to return a set of genomic coordinates that can help inform the selection of a correct transcript. In part 2, we actually compute that reference sequence using the CoolSeq tool or common operations of the CoolSeq tool to basically determine the correct sequence and then also compute an offset. So if you think about the target sequence being like a substring of a human reference sequence, we'll have to kind of describe those variants with respect to that offset. So that's important data for mapping it correctly to a human reference sequence. And in part 3, we standardize MAVEDB variants. And to this, we use the variation representation specification of the Global Alliance for Genomics and Health. It's basically a standardized way to represent variants in a machine-readable format. We also use the Biocommon State Repo database, which is a database for biological sequences that can be downloaded locally and modified. And once this step is done, we have MAVE data that's available for downstream integration. So for step 1, we align the target sequence for a score set to GRCH38 using BLAT. We select the best hit for a score set and store in a dictionary. So in this example, we have score set 41A1, we record metadata including the chromosome, the strain orientation, the target name, target type, uniprote accession, and kind of metrics of coverage and identity. But the main thing is that in the red box, the hit ranges, which correspond to the genomic coordinates for this target sequence. So here we have 6 different kind of blocks that correspond to the target sequence for how it aligns to the human genome. This kind of second step where we select the reference sequence, so this is kind of a visualization of that example that I showed in the previous slide. So suppose we have 3 different transcripts, B, R, and G. B corresponds to the main select transcript, red to the main plus clinical, and gray for other possible transcripts. So for the first chunk, we have all 3 transcripts kind of overlap with that position. So we report BRG, same for block 2, but for block 3 we only have R and G, and then for blocks 4, 5, and 6 we have BR and G. So if we intersect all those lists, we see that we have R and G in all 6 positions, and then kind of following prioritization of the main transcript over the other compatible transcripts, we select the main plus clinical as our transcript, and then compute an offset if we determine that the target sequence is a substring of the overall reference sequence. The last step is once we have this kind of reference sequence determined and the offset computed, we want to standardize the MAVE variants. So first we kind of register the target sequences with seq repo locally. So as you can imagine, these target sequences could be mutagenized or not correspond to the human reference sequence. So we need to have these kind of installed locally in seq repo to enable normalization of those variants. So we have the same kind of mapping for both the pre-map variant and the post-map variant. And then once we kind of have those sequences added to seq repo, we create a representation for the assayed or pre-map variant. And then we update the sequence location using the offset that we compute in part 2 for the map variant, and then we basically add to a dictionary. And these assayed variants and map variants are homologous variants, and we leverage the sequence ontology, in particular the homologous to relation, to compare these variants, as they're not necessarily equivalent, but correspond to similar locations on the examined sequences. So after completing this process, we have mappings complete for 207 score sets in MAVE-DB, and this encompasses roughly 2.5 million protein and genomic variants across the examined score sets. And the question that emerges from this point is how we assess accuracy. And I think a good metric to do this is by basically assessing whether or not the reference sequence is matched for each allele pair. So if we have an assayed variant and a map variant, does the reference sequence in that position agree for both the assayed variant and the map variant? And what we found is a 99% overall reference sequence match between the pre-mapped and post-map variants. And there are 2 main causes of discordance across these remaining 1% of variants that didn't have complete agreement in the reference sequence match. The first cause you can imagine is if you have variants that kind of span intron-exon boundaries and are multinucleotide variations, such as deletions, insertions, or deletions, insertions. So in the target sequence on the left, suppose we have a deletion that spans 3 base pairs corresponding from the start to end position. But if that start position aligns on one exon and the end on another exon, and those are spread out over thousands of nucleotides, it's hard to create a consistent computable object to describe that variation when we have the variants kind of occurring over 2 large chunks in the human genome. A similar issue occurs for when we have a mutagenized target sequence, where suppose we have complete equivalency for all positions but one, which is the G for the target sequence, and we have a C in the human reference sequence. So we could have a substitution at that position for the target sequence, which would disagree with the human reference sequence position. So those are kind of the 2 main causes of discordance that we saw when trying to map these variants and trying to compute the accuracy of the mappings. This effort enables dissemination of MAVE data to different downstream resources. One resource is the Genomics of Protein portal from the Broad. So this is a tool to link genetic screening outputs to protein variation. And as you can see on the bottom, in addition to kind of relevant protein data, we're able to show data from different MAVEs for the protein of interest. So we had this complete for, I think, 168 protein coding score sets. So yeah. Another integration is the UCSC genome browser. So we have MAVE score set data available as a track hub in the genome browser. So in this example, we have at all possible positions a variant and different colors that kind of correspond to the enrichment score at that position. So basically, a user could kind of add this annotation data to their existing data they're looking at in the browser when they're trying to interrogate variants of interest and assess their clinical relevance. Lastly, we have MAVE data available in the ensemble variant effect predictor. So this is kind of a new column that you can see when querying a variant of interest. So we have two variants, two missense variants in UBE2I, and we're able to kind of report this MAVE-DB score that can be used to help clinical interpretation by providing additional functional effect data that was not previously present. Lastly, in the remaining time, I'd like to go over the MEMA information model. So this has been done in collaboration with the AVE Alliance. So basically, we're defining a minimum standard for how to report MAVE variants. Some useful fields include the project summary, the experiment summary, the library generation method, the cellular context, the phenotypic assay, and the sequencing techniques that were used to generate that MAVE variant. We also encourage users to upload their sequence metadata to MAVE-DB and seq repo when uploading new score sets to MAVE-DB, as it can help facilitate mapping of the data. Lastly, for conclusions, MAVE data can be mapped to human reference sequences, and the next step I see is kind of calibrating this MAVE data for clinical interpretation. And this is an effort undergoing in the AVE clinical variant interpretation workstream to make this data more readily available for downstream clinical interpretation. If you're interested in this work, I encourage you to read our manuscript on bioRxiv, or preprint on bioRxiv. I'd like to acknowledge my lab, Nationwide Children's Hospital, Ohio State, AVE, and CGC for the travel fellowship, and I'm happy to take any questions. Thank you.